They'll be in lecture group 1 when it does come up, or they'll be in part 2?
They will be in the folder, and they will be part 2. Which brings up another point. So lectures 1 through 9 that we have done so far, including today so far, that's what's on exam 1. So exam 10 and beyond is going to be, or sorry, lecture 10 and beyond. So this material and beyond will be for exam 2. So the second half is no question.
Yeah, I had a question about the mosaic development. So
Okay.
can you not have monozygotic identical twins in mosaic development?
I don't understand the question. You do not have monozygotic.
Twins, yeah, in like an embryo that is using mosaic development.
So if we had, say, C. elegans, could you get monozygotic identical twins? I think the answer would be probably not, because let's say that you had a C. elegans embryo that separated into 2 clumps, they would not be able to do regulative development, and they would not therefore make 2 complete individuals. So actually that's a nice example, that's a nice question to ask, because I think it illustrates that difference. Does that make sense? Okay, so if you have C. elegans where you have autonomous lineage-based factors controlling these cells, you separate them into 2 clumps. These now are missing some of the cell fates and cannot reestablish themselves as separate individuals, which is what you would need to get the twins, right? But in the case of humans that could do it by regulative development, they can adjust, because it's not based on lineage, it's based on cell interactions. So within each clump, the cells interact, they figure it out, they establish all the cell fates, and therefore can make 2 nice individuals.
Going back to the invariant cell lineage, is that just for like the hermaphrodites, or does it apply for both the hermaphrodites and the male sexes?
The invariant cell lineage also applies to the males.
Okay.
So they have different genetics in terms
Sure.
of chromosomes and so forth, and so that's going to play out in terms of what their ultimate morphology looks like.
Right.
But the mechanism of separating the cells in a specific fashion and the same mode of development all works in the male individuals too.
Okay.
Yeah, good question. Any other thoughts? Yeah.
So does the sperm on the C. elegans have to fertilize in that home location? In other words, could it physically go through any part of the shell? Is the first question. And then the second part is, if it did go somewhere else, does it develop normally, or is it because that establishes the axis and not develop normally?
I see. So could this sperm be used anywhere else on the egg cell
Yeah.
membrane, and if that did happen, would it be normal? So I don't actually know the answer to that, so I'm going to give you what I think. I think that it would not usually fertilize anywhere else because it simply can't get to it, but that if it could get to it, it would fertilize there successfully. So then you would reestablish the zygote. And then I would see that male proteoglys is coming in to that cytoplasm in an abnormal location. And it would depend on if that embryo can still sort this out to get that cleavage plane back to the center of the egg, or like offset center a little bit. If it can do that, then it would divide the cytoplasm normally, and then from that point on, the embryo would be normal. But if that original mitotic spindle is set up anywhere differently within that original cytoplasm, then you're going to influence how that would proceed into the 2 daughters, and if that's not normal, then the embryo will be normal. Okay, so now we're going to talk about Drosophila, and this is actually, these next few lectures are kind of some of my favorite material for the course. I just think it's kind of neat how we were able to start with the genetic screen, find these genes, see what they do, and really learn tremendous about how this happens in Drosophila. So here's an introduction to the Drosophila system. The main feature about Drosophila that it was always well known for the last 100 years is its genetics. And this is a really well understood genetic system. It is basically diploid Mendelian genetics, but there's all sorts of tools and possibilities for manipulating it, all kinds of different mutant constructs out there, and just a really nice workable system to get deep into the genetics and figure out a lot of stuff. It's also really easy to raise these in the lab. All you need is a milk bottle with some glucose-based agar and a piece of tissue for the flies to crawl up and do metamorphosis on. They're dead cheap. They are lots of different kinds of cell fates and tissues and eyes and organs and bristles, so lots of things to mutate and figure out, what the genes are for this. So the Drosophila life cycle takes about 10 days to go from a fertilized egg. You're going to go through some embryonic period, 3 different molting stages. The third larvae that is developed here will then crawl up out of the food mass onto a substrate such as the wall of your bottle or a piece of tissue and form a pupa. And inside the pupa they'll go through metamorphosis, and out pops the amazing adult fly. So 10 days to do all this, which is quite a reasonable short period of time, still workable for doing multiple generations of crosses. Okay. So that's the fruit fly. And here's just a little snapshot of the reproductive systems in the female. We have a uterus, we have an oviduct, and then 2 ovaries up on top that are responsible for making eggs. And as these eggs develop, they'll slide down the uterus and into the ovary. And along the way, they're going to pass these 2 little tubules that have sperm stored in them. So males, when they mate with the females, will deposit a big bolus of sperm. The female then takes these sperms and puts it in these specialized organs that can maintain the sperm alive. And then eggs go by, a sperm comes out, and this enables the female to fertilize one female, say, 2 or more progeny just from a single mating. So you can get a lot of offspring from fruit flies. And that's also quite handy if you're trying to do genetics, right? You might be looking for a small mutant class with a particular genotype, but you can have no trouble to get these due to the large number of offspring. Okay, here's just a snapshot inside of some of these sperm storage organs. Up there you can see some very long, thin marks. Those are the compacted sperm nuclei that are being stained inside of the tubule that the female reproductive system has for storing sperm. Okay, so here's another peeling back up here on the ovary. You can see these strands of chambers. Each of these round things is called an egg chamber. And we're seeing in here one of the cells is the haploid oocyte that will give rise to the zygote. And then a number of other cells associated with this that are called nurse cells. And these nurse cells make a number of products that they then transfer over to the egg and put it inside the egg. So all these maternal factors like RNAs and proteins are actually synthesized in the nurse cells. And then there are these special little channels where these factors can be transported and then dumped right into the oocyte. Then there's another layer of cells here in purple called follicle cells, and these are also going to be important for some of the patterning events in the oocyte. This is the mature egg. It has these 2 little flaps, appendages that poke out of the dirt or soil or agar and help the respiration or breathing of that cell. Okay, so within these egg chambers, as we said, the diploid nurse cells are making products, RNAs and proteins, and transferring them on over into the oocyte. Once they get into the oocyte, they are not symmetrically distributed evenly across the oocyte, but often become sequestered into one corner or one portion of that oocyte. So this is a similar sort of thing with the P granules. You're not having that factor just be anywhere and everywhere, but in fact it's localized to some nice little nifty corner. That then becomes important as that oocyte gets fertilized and goes on through later development. So in this case, we're looking here at the early oocyte shown in yellow, The follicle cells shown in dark gray and light gray, and then these big nurse cells here with the blue nuclei. And I want to point out a few of the features. Initially, within the oocyte, the nucleus is down towards the posterior end. That is the side further away from the nerve cells. And there's some communication going on with the overlying follicle cells. So these follicle cells have a receptor that's called a torpedo, shown here as a little orange Y-shaped thing. And then the nucleus is making this protein called gherkin. So these two are talking back and forth and starting to establish some polarity to this egg. So that's going to be the posterior side there. Then as time goes by, this oocyte's getting a little bit bigger, and we're also building an array of microtubules. And these microtubules are oriented all in the same way, with their minus ends closer to the nerve cells, and the plus ends closer to this posterior side for the oocyte. And then as maternal components get dumped into that oocyte, oftentimes there'll be these molecular motors, the dynein and the kinesins, that are attached to the microtubules, will grab these RNAs and then transfer them over to one portion of the embryo. So for example, we're going to have some dynein and kinesin. Let's get that over. Yeah, okay, so here's the example of up on the top. We have a dynein molecule with a little red jelly bean there. It's picked up a bicoid mRNA, so that's a very specific gene product. And bicoid mRNAs are being carried towards the minus end of these microtubules. So all the bicoid mRNAs are going to end up localized right here, right next to the nerve cells at the future anterior side of this oocyte. In the other case, we have some kinesin that's grabbed a different mRNA, Oscar, and is carrying that down to the posterior side. And this becomes actually critical for the future patterning of the embryo once it's fertilized. So they're very specific in terms of what cargo and where it goes. So you can tell this experimentally by doing experiments like in situ hybridization to ask, where is that bicoid mRNA in the embryo? And you find out, oh, all signal for this this particular mRNA is located right here at the anterior side of this oocyte in the egg chamber. Okay, so already by mid-oogenesis, a lot of patterning has happened and these have become sequestered. Okay, here's just to show you, so this makes a little bit more sense. As time, the oocyte's going to get bigger and bigger and bigger, and eventually the nerve cells are going to kind of degrade. So initially the figures will look like this with the oocyte here on one side and nerve cells, and then eventually the oocyte gets really big, and this is the remnant of the nerve cells up here. And you still have a layer of cells called follicle cells surrounding this whole thing. Okay, so after fertilization, what happens in Drosophila is that original diploid nucleus will go through mitosis and start replicating itself to produce daughter nuclei. However, you do not get karyokinesis and cytokinesis at the same time here. So karyokinesis is the replication and separation of nuclei. That happens. But cytokinesis, which is the dividing up of cytoplasm into daughter cells surrounded by membrane, that is temporarily on hold. So we did get here in this common cytoplasm of the original fertilized oocyte, nuclei going through mitosis. And they're stained here with DAPI, so they're yet white in color. And initially these nuclei are just all distributed together in a common cytoplasm. Then what we'll see is around cycle 10 or so, the 10th cell division, these nuclei change their location from being interior where all the yolk is. They move up to the periphery and then become arrayed all around the outside periphery of this embryo. And then we're going to see that cell membrane folds down in between each of these nuclei and starts to pinch off now complete cells surrounded by plasma membrane. So you have now for the first time an actual layer of cells with a nuclei, cytoplasm, and cell membrane going around the entire cell. So initially, we call this the syncytial blastoderm because the nuclei are in a syncytium, they're there together. And then later we call it a cellular blastoderm because the nuclei are encompassed within individual cells. I'll just point out the pole cells here. These are the very first cells in the embryo that acquire their own private cell membrane. And these are going to become the germ cells for Drosophila. And so they're just sequestered very early in development as being special group of cells. So Drosophila, during all of these initial divisions, it's before the mid-blastular transition. And what we see is all these nuclei tend to divide at the same time. They're all in synchrony. And you can make these beautiful time-lapse movies using different sorts of markers for DNA or tubulin along the outside of the cell wall and track how these things are going through mitosis. And it's just amazing to see all of these nuclei divide at the same time, and they pause, they all divide again, and then they begin to relocalize their position up here to the periphery. So then after the mid-blastular transition, this will kind of be less true, that they will now be more asynchronous. Of course, they'll be then beginning to transcribe their own RNAs, and some will be dividing rapidly and some will be dividing slowly, and development goes on from there. So looking a little bit more at the events of actual cellularization, once these nuclei move up to the surface, each nucleus is associated with a centrosome or a microtubule organizing center. You of course need this in order to go through mitosis and set up a mitotic spindle. But they also use the centriole to help themselves, to help these nuclei attach to the plasma membrane up here at the periphery. So they're using microtubules to help anchor these nuclei up at the cell periphery. Then in black here you see the plasma membrane, and it's starting to fold in and grow down in between each individual nucleus. And microfilaments, that is an actin-based macromolecule. These are going to be important for stabilizing those furrows, so there's called furrow canals. These will be stabilized through the presence of actin microfilaments. And then a little bit later down, these furrows will come all the way down past the nuclei, and laterally they'll start to seal off a layer of cells here up at the periphery. All the yolk of the Drosophila embryo is now in the middle, not really in the cells. So you can also see this by using fluorescent markers for things like tubulin or actin, and see how these cells are forming, and look at the timing and speed of all of this. Okay, so here you can see just the completion of a dot where they've now formed an entire single cell layer up at the periphery. And before the mid-blastular transition, these waves of synchronous division spreading across the cells, embryos. Okay? Then after we get through the initial cleavage process, just like for other organisms, our next goal is vasculation. Drosophila do this a little differently than frogs. They have 3 places where you will see involution, cells on the outside migrating inward towards the middle. This starts on one side of the embryo with a furrow, just invaginating or folding inward along the anterior posterior axis. So this starts on the ventral side, and it's called the ventral furrow, where cells are invaginating into the interior of the embryo. And eventually then that will seal shut, giving you some cells inside and then some cells left on the outside surface. If you look at that in cross-section, you can see these cells labeled with the brown, so it's some gene being expressed in those particular cells. As they begin to fold inward, now these brown labeled cells will become interiorized to the embryo, and the furrow will seal shut on the outside. cells in two other places. Up in the anterior is another site of just invagination where a group of cells will enter the interior. And then down here at the posterior, the pole cells, those very first cells that became true cells, they also get folded inward into the interior of the embryo. And then we also have this thing called the cephalic furrow, which is just an indentation that sort of denotes where the head is versus the rest of the body. That's not a site of gastrulation, but it's a landmark morphologically that we can see in embryos. Okay, after gastrulation, this initial kind of log-shaped or rod-shaped structure begins to do some interesting things morphologically. It begins to separate into these segments. You can truly see here some morphologically distinct regions within this embryo